The chemical senses. The senses of smell and taste are called chemical senses because they extract information from the environment by means of direct chemical interactions. Molecules from external substances interact with receptor molecules in the nose and the mouth, resulting in the generation of neural signals. Chemical molecules are weighted to olfactory chemoreceptors in the nose by atmospheric currents, so the stimulating substance itself can be quite remote from the perceiver. Taste, on the other hand, is a contact sense. Molecules have to be in a solution that makes direct contact with the chemoreceptors. We will learn that each sense will be considered first in terms of its anatomy and physiology, and second in terms of its perceptual and functional properties. Clear relationships between anatomy and physiology on the one hand, and perceptual and functional properties on the other, will emerge. First, we have smell and the anatomy and physiology of smell. You have certain olfactory receptor neurons, and they are found in the roof of the nasal cavity on a patch of tissue called the olfactory epithelium. And of course, we also are talking about free nerve endings, which are branches of a sensory nerve cell that has no specialized receptor process but is embedded directly in tissue. We have the olfactory receptor neuron, which is a specialized neuron that produces electrical responses to odor molecules. We also have the olfactory epithelium, which I just mentioned, which is a patch of mucous membrane in the roof of the nasal cavity, one for each nostril, and it contains olfactory receptor neurons. In humans, it contains 6 million receptor neurons. Okay, so because our sense of smell is blunted when we have a cold, and because of the buildup of mucus in the nasal cavity, that prevents odor molecules that reach the receptor cilia. And of course, we might talk about the mitral cell, which is a neuron in the olfactory bulb that receives signals from olfactory receptor neurons, and it also relays them to the brain. There are 50,000 mitral cells in the human olfactory bulb. The olfactory bulb is the mass of neuronal tissue protruding from the brain behind the nose, which conveys neural signals from the olfactory epithelium to the brain. There's the olfactory glomerulus, which is a dense spherical accumulation of dendrites and synapses, where approximately 200 olfactory receptors make contact with a single mitral cell. Then there's the primary olfactory cortex, which is the cortical destination of mitral cell fibers, thought to mediate perception of smell. And then we have the interesting amygdala, which is a nucleus, a dense group of neurons which lie deep in the brain. They form part of the limbic system, and they are involved in emotional, sexual, and automatic responses. For example, we have sensory pathways, these cortical processing. We have the perception and function of smell. First we have detection, and then we have recognition. And so there's all kinds of odor recognitions. Odor is identified most reliably, for example, by the chemical senses. Then we have from adaptation as well. Adaptation is something that's happened from an evolutionary perspective, evolutionary psychology's perspective. In insomnia, we have that, which is anosmia, and anosmia, which is odor blindness. And then of course things like that, like we have pleasantness and unpleasantness, the function and role of smell, or edible or inedible, which would have helped our prehistoric ancestors. We also have taste, the anatomy and physiology of taste. We have taste receptors, which are chemoreceptor cells found on taste buds in the mouth, tongue, and throat. There are 50 to 150 receptors on each bud of the tongue. Of course, a taste bud is a cluster of cells embedded in the skin of the tongue and the mouth, housing taste receptors. The human tongue contains approximately 5,000 taste buds. And the gustatory papilla is a small elevation in the tongue, which is visible to the naked eye, and it contains up to several hundred taste buds. And the umami, which is a Japanese word meaning good taste, and recent evidence indicates that it is a fifth basic taste quality. We have, obviously, taste adaptation as well, sensory pathways and cortical processing, and the anatomy and physiology of taste. And of course, taste primaries is another thing, which is a theory of taste coding based on the idea that perceived taste can be broken down into basic qualities, each uniquely associated with activity in a specific neural pathway. Then we get to, for example, we're thinking about the taste system seems to function as a nutritional gatekeeper, regulating consumption and rejection on the basis of edibility and nutritional value, as signaled by the 5 taste qualities of sweetness, saltiness, sourness, bitterness, and savoriness. And I won't go into all of that. Of course, they have an entire evaluation of flavor, for example, in the culinary sciences and things like that. So, again, we'll talk about how we'll reflect the general principles that we discussed earlier and how they relate specifically to the chemical senses. You have qualia and modality. The smells and tastes directed by the receptors generate powerful sensory states, or qualia. The clean smells of limes, the yumminess in foods containing MSG, the disgusting odor of feces. In some respects, the two senses operate as distinct modalities, but they are also tightly integrated to create flavors that blend the two senses together. And you have psychophysics, which we've discussed earlier. We are incredibly sensitive to certain smells, but thresholds vary markedly from one odorant to another. The detectability, however, of olfactory and gustatory stimuli varies markedly over time and helps us to adapt to specific environments in terms of our chemical senses. The taste primaries theory is based on a clearly articulated psychophysical linking hypothesis, which proposes a direct relation between activity and sensory nerves and specific experiences. Then we have what's called cognitive neuroscience. Both smell and taste are based on chemoreceptors, which show a clear preference for particular odorants, or taste, for example, but both senses also exhibit a clear hierarchy of processing that begins at the receptors in the nose, mouth, and throat, and of course ends with activity in the sensory cortex, passing through intermediate stages on the way. And of course, topographic maps feature in both systems. And then computational neuroscience. There are very good examples of representation in the chemical senses. In the case of smell, recall that any one receptor neuron responds to multiple odorants, and any one odorant excites multiple receptor neurons. And so it follows that different odorants excite different combinations of receptor neurons. And so we can therefore consider the pattern of activity across the population of receptor neuron types as representing the unique chemical composition of the odorant. If the odorant composition changes, the pattern of activity, which is representational state, and the receptors also change. The pattern of activity actually forms a spatial representation of the odorant by virtue of the spatial arrangement of glomeruli in the olfactory bulb. For example, we might propose that the pattern of activity constitutes a combinatorial code for odor. There are roughly 1,000 different receptor neuron types. And even if we do assume that just 3 types respond to each odor, the code would be able to discriminate nearly 1 billion different odors. And of course, in other words, it would have nearly 1 billion different representational states. And so in the case of taste, receptors fall into just 5 classes, responding optimally to sugar, salts, acids, which are sours, and plant alkaloids, which are bitter, and glutamic acid, umami. But each activity in each receptor class appears to represent the presence of the correspondent substance. A specific state in the sensory system maps into a specific state in the outside world, the presence of a particular kind of substance. The psychophysical and neural characteristics of the chemical senses allow us to infer what kinds of computations they perform: approach avoidance, edibility, and nutritional value. And again, we're talking about the chemical senses. So we're going to go a little bit into smell, okay? Senses of smell and taste are known as chemical senses because their receptor neurons extract information by means of chemical interactions. So we have this smell. The sense of smell is mediated by 500 to 1,000 different types of sensory receptor in the olfactory epithelium of the nose. Each mitral cell in the olfactory bulb of the brain is connected to just one type of receptor neuron. Different environmental substances create different patterns of activity across the mitral cells. The ability of humans to recognize thousands of different odors depends on the ability of the olfactory sensory system to distinguish different patterns of activity in mitral cells. The large number of partial anosmias found in humans is a reflection of the relatively large number of different olfactory receptors present in the sensory system, and the function of smell is to act as an early warning system, which classifies smells in terms of two dimensions: approach avoidance and substance edibility. Concerning taste, all taste can be decomposed into contributions from 5 primary qualities: sweetness, saltiness, sourness, bitterness, and savoriness, or umami. And again, just go over what the hell umami is. Well, I don't really know. I'll find out. Go read it and find out. But anyway, so gustatory receptors fall into 5 categories on the basis of their preference for one or 5 types of substance, namely sugars, salts, acids, plant alkaloids, and amino acids. Of course, these receptor neuron types map precisely onto the primary taste qualities identified perceptually. The taste primaries theory of taste coding proposes that the receptor neuron types are labeled lines coding the presence of specific substances. The properties of the sensory pathway and cortical representation of the taste are consistent with the taste primaries theory. Of course, the function of the gustatory system is to detect different types of important substance because there is adaptive value in regulating their intake, consuming some and avoiding others. In terms of flavor, food flavor during eating involves an interaction between several perceptual sensations, including taste, smell, temperature, touch, sight, sound, and pain. Cells responsive to combinations of smell, taste, and sight may mediate flavor sensations. And so we have a smell and emotion, okay? Certain smells that enhance attractiveness dates all the way back to the beginning of our recorded history, for example, in the cultures of the Middle East. But anyway, we're talking, and also there's a variety of chemical receptors. For example, the chemical senses are the most primitive of all the senses from an evolutionary perspective. You're thinking about chemosensitivity. It undoubtedly emerged very early in the history of life on Earth, and primitive bacteria immersed in a watery medium awash with chemicals. Of course, anyways. And so there's a lot of things that we can look about the chemoreceptors found in the human gustatory and olfactory senses, and this represents just a small part of a broad spectrum of chemoreceptors that serve a number of different functions in different organisms. And so next we're going to go to the body senses. We just covered the chemical senses, which are the most basic senses that a creature or a living thing can have. Next we'll go to the body senses. Not every living thing, most things have a body, but I guess cells, they still have a cell body. So we're going to get to the somatosensory system. Okay, we're going to go to that, which is more touch. You know, it's called somatosensation, kinesthesis, nociception, vestibular sense, and we're going to go to proprioception as well. So we're going to talk about all these different terms in the body senses. We're going to talk about the all these different types of perception and a somatosensory system, the vestibular system, and things of that nature.